Hello students. In this video I am discussing about the enzymes. Enzymes are catalysts. Catalysts are those substances which they take part in the reaction but they themselves are not altered and they help in carrying out the reaction at a very high speed. The rate of reaction increases. Such molecules are called catalysts. Let us see the definition. Substances that have capacity to speed up the reaction without themselves being altered after the reaction are called catalysts. Now, because they are again generated back from the reaction, they are required in very small quantity. And these enzymes, when there are chemical catalysts also which are used in the laboratory. In the laboratory, when these catalysts are used, in the test tubes, then at that point the temperature may vary or the pH of the chemical may vary. That will not create any difference, that will not matter a lot. But inside the body, when there is controlled temperature, controlled pH, and in that controlled environment, when so many reactions are being carried out in the body, then enzymes are the catalysts. Those catalysts are called enzymes. They are organic catalysts. They are produced in the living cell and they are highly specific in their function, and they can carry out this or the speed up the reaction or carry out the reaction at a body temperature and a given pH in an effective and optimal manner. So enzymes play a very important role because they carry out the reactions of the body at optimal temperature and pH inside our body. Now these enzymes are proteins. They are proteins, and how can we tell that they are proteins? The characteristics of proteins are they get denatured by heat, they precipitate by ethanol, and they do not pass through semi-permeable membrane. So all the enzymes, they execute this property, and hence all the enzymes are proteins. Now enzymes, they are composed of two parts, coenzyme and apoenzyme. They both combine to form the holoenzyme, and holoenzyme is the active enzyme. A coenzyme is inactive, low molecular weight organic molecule which is dialysable. And apoenzyme are inactive, high molecular weight and non-dialysable. These two components will combine to form holoenzyme. Let us see the classification of enzymes. Classification of enzymes. Enzymes are classified into 6 groups. First group is oxidoreductase. Oxido means oxidation, rid means reduction, reactions. The enzymes which help to carry out the oxidation reduction reaction are oxidoreductases. Here I have written electron transfer reactions. They help in electron transfer reactions. They transfer the electron or hydrogen ion. As we know that oxidation is removal of electrons and reduction is addition of hydrogen ions. So they carry out transfer of electrons or hydrogen ions. Next is transferases. Transferases are those enzymes which transfer the functional groups. Phosphate group, amino group, methyl group, etc. They get transferred onto a molecule by the help of transferases. Next category of enzymes are hydrolases. Hydrolysis reaction. They carry out the hydrolysis reaction, means addition of water molecule to break a bond. Wherever the bond, double bond is there, that is broken, and a water molecule is added at the place of broken double bond. Such reactions are carried out by hydrolases. Next is lyasis. Lyasis is addition at a double bond in a molecule. This is non-hydrolytic. Water molecules is not added while the double bond is created. Next is isomerases. This enzymes, they create isomers of a molecule. Isomers have same molecular formula, but their orientation of molecule is different inside the molecule. So such are isomers. So they are carried out by isomerases. Then next is ligases. Ligases reactions, they are formation of bond with cleavage or breakage of ATP. By using the ATP, the bond is broken. That is carried out by ligases. Next is condition that affect the activity of enzyme. The conditions which affect the activity of enzymes are concentration of enzymes, concentration of substrate, pH and temperature. As I have said that enzymes are very specific molecules which carry out the reactions at body temperature and body pH. So for the functioning of the enzymes, pH and temperature play a very important role for optimal activity of the enzyme. Next is mechanism of enzymes. All enzymes have active sites and the substrates have affinity for the active site. Substrate, they gets attached to the active sites and they form an intermediate complex, and from that the enzymes will either add a group or remove a group or add a double bond at that site, and the final product will be released. So uh, main function of enzyme is to lower the activation energy. The amount of energy required to bring a substrate to a reactive site is called activation energy, and this enzyme lowers the activation energy. So at a very low energy, lots of substrates are converted into the products. So this is how the reaction takes place. Enzymes plus substrate will form an enzyme substrate complex. This is a transition state, requires less activation energy. And from this complex, then end products plus free enzymes are released. So this is outline of how enzyme substrate reactions are taken